students. Today we are studying about brassinosteroids. It's a type of growth regulators found in plants for the cell expansion and their development. Steroid hormones have long been known in animals, but they have only recently been discovered in plants. Animal steroid hormones include the sex hormone like estrogens, androgens, and progestins, and the adrenal cortex hormone like glucocorticoids, mineralocorticoids, etc. The brassinosteroids are a group of steroid hormones that play a vital role in a wide range of developmental phenomena in plants, including cell division and cell elongation in stems and roots, photomorphogenesis, reproductive development, leaf senescence, and stress responses. The identification of plant steroid hormones was the result of nearly 30 years of efforts to identify novel growth promoting substances in pollen from many different plant species. Early studies by J. W. Michel and colleagues showed that the greatest growth stimulating activity was found in the organic solvent extract of pollen from the rape plant. The unidentified active compounds in rape pollen were named brassins. The specific growth promoting effects of the brassins were scored in several physiological tests, including the Bean second internode biopsy. In this assay, brassins behave differently than other known phytohormones, it causing both cell elongation and cell division, as well as bending, swelling, and splitting of the second internode. Brassinosteroids structure, occurrence, and genetic analysis. Two main bioassays have been used to purify brassinosteroids: the Bean second internode assay and the Rice lamina inclination bioassay. These bioassays distinguish between biological active brassinosteroids and their inactive intermediates or metabolism and allow quantification of the amount of active compounds which are present. The basic chemical structure of a brassinosteroid was the first resolved in 1979 when X-ray crystallographic analysis of the purified active substances determined that it was a steroidal lactone. The compound was named brassinoloid, and its characterization ultimately led to the chemical identification of a group of about 60 related phytosteroids called brassinosteroids. The direct biosynthetic precursor of BL testosterone has weak brassinosteroids activity as well. Brassinosteroids have now been identified in 27 families of seed plants, including both angiosperms and gymnosperm, and one is pteridophyte, which is equisetum. One bryophyte like Markensia polymorpha, and one green alga, Hydrodiction reticulatum. Thus, Brassinosteroids appear to be ubiquitous plant hormones that predate the evolution of land plants. In the angiosperms, brassinosteroids are found at low levels in pollens, anthers, seeds, leaves, stems, root, flower, and young vegetative tissues. Knowing the molecular structure of BL allowed investigators to synthesize both naturally occurring brassinosteroids and their related analogues. By testing these compounds with either the bean's second internode or rice lamina inclination biopsy, the following key requirements for BR activity were determined. First is a cis-vicinal glycol function at C2 and C3 of ring A. The absence of a single hydroxyl group at either C2 or C3 or any change in their configuration results in a significant loss of function. Next is the 7-membered B ring lactone, although limited modifications of the B ring don't eliminate activity, they significantly reduce activity. The steroid side chain with hydroxyl group at C22 and C23. The alpha orientation at C22 and C23 confers higher activity than the corresponding beta orientation. Here is a figure: dwarf lamina, rice lamina inclination biopsy for brassinosteroids. A small droplet of sample brassinosteroid dissolved in ethanol is applied to the joint between the lamina and the leaf sheet. After incubation for 2 days in high humidity, the bending angle theta between the lamina and the leaf sheet is measured. The angle is proportional to the amount of brassinosteroid in the sample. So it's a biopsy method for brassinosteroids, which is called dwarf rice lamina inclination biopsy method. So आज के लिए इतना ही. थैंक यू स्टूडेंट्स.